Today we learn about designing primer. Primer is used in polymerase chain reaction for the amplification of DNA. First of all, what is primer? So primer is a short 1824 base pair oligonucleotide sequence used in PCR. There are many paired and unpaired software available for the designing PCR primer. Today I will show you how to design primer in Primer 3 and Primer 3 Plus website. These two are more cited and more performed in research primer designing. So let's get started.
First of all, you will open four websites: Ensemble Genome Browser, Primer 3, and Primer 3 Plus, and in silico PCR. All these website links are in the description. You can click in the description. These websites, all the links are in the description. These website links are in the description below, so you can check in the description. And then in Ensemble you can search your desired of gene, which genes you want to primer make. So I'll search HBB gene, and then find in human HBB gene. In human, it is a human gene, hemoglobin beta. So then step by step process you can follow me on this procedure. You can click on a sequence, and then you can find here in symbol the exon and intron region of that sequence by transcript id, and ccd s is the id of uniprot, which having all the nucleotide and protein sequence in that. Then here you can find the exon and intron region, and just you will find the first exon of that sequence. So you can copy some of the bases of that sequence, and then control F, find in that region. So these are the exon region. Here are the exons of that gene in the genome, chromosome number 11. You can copy it, some sequence, and paste in that region where you can find. So after that, when you find the region of the first exon, then you will copy the complete region of that sequence. After that, you copy that region where have your desired gene, and then paste it into the Word file or any text note. So first here there is your sequence and your desired sequence too, and you have to find the primer in that regions. So before you paste in the primer 3, you will select the region. So after some 14-15 nucleotide before you will uh, enter, you will write a bracket, and at the last of the first exon you will also select a bracket to software will detect that region. I that region the algorithms are sit in the software. So these bracket will detect that the person need it at that region primer. So then copy that sequence and paste it into the primer 3 plus. Note that your target region will not more than 500 to 1000 base pair. You will check into the words numbering, that how much is your desired region bases. So then copy that region and paste it into the primer 3 plus. And in primer 3 plus you can change the tm and enabling temperature and primer size, and just pick the primer, and they will show your desired sequence in a greater than sign or smaller than sign, I think. And the your primer will be shaded. In the primer 3, after that you can copy the forward primer and reverse primer and put into the in silico PCR to detect that as my primer are exactly A for the chromosome number 11 at some region or not. So it will show you it will show you the confirmation of your sequence that your primers are right or wrong. So and this is the primer 3 plus, so just paste the sequence and pick primer and it will give you in a shaded form your sequence in green and some other colors, your power primer and reverse primer, and copy that sequence into the in silico PCR that will confirm your region and that will be your primer. Hope you will learn. If any queries, please comment.